Remember, GI Packet 3 will be a video, and we will not have live class on Monday. So we left off looking at the mechanisms by which our macromolecules were digested into their smaller components and then absorbed across the epithelium as those micromolecules or micronutrients. And we left off here looking at proteins. So we spoke about how carbs are digested. We spoke about some of those transport mechanisms, active transport, facilitated diffusion. And we're gonna move on here to look at how proteins are absorbed. So proteins are broken down by proteases, and this is just a general class of molecules. They're enzymes, to be specific, that break down proteins. There are many different types of proteases in terms of classifying them as to how they break down those proteins, as well as the specific type of protein they break down. So we've got endopeptidases. These split polypeptides somewhere in between the peptide bond. So they're not going to be cleaving out at the periphery, but these proteases cleave specifically within the peptide fragments. We've also got exopeptidases, which cleave out to the periphery. So these are going to cleave amino acids that are on the ends of these polypeptide chains. And then we have a general class of enzymes that we call zymogens, which are the inactive storage form of these proteases. So zymogens are basically these inactive enzymes that are stored in the pancreas for the most part. They're stored in these granules called zymogen granules. And they are secreted into the lumen via exocytosis. And these make their way to the GI, to the duodenum specifically, from the pancreas where they're stored. And they are then activated, converted to their activated form, and then they can begin to break down the protein. So the way that we store these enzymes is in an inactivated form. They're generally called zymogens. And then they can be activated when they get to the lumen of the duodenum, and then they can begin to break down these proteins, either cleaving the amino acids at the periphery, if they're exopeptidases, or cleaving amino acids within the peptide bond if they are endopeptidases. Let's look specifically at pepsin. So pepsin is just one type of protease. So don't let the names kind of confuse you here. So one example of a protease is pepsin. And pepsin is actually found in the stomach. And so this is where protein digestion begins. Pepsin is released from these gastric pits. So if you remember our last class, we spoke about the stomach having these pits or these invaginations that we call gastric pits. And down within these pits, we had different types of cells that secrete different substances onto the lumen of the stomach. So we had chief cells, we had parietal cells. Chief cells secrete pepsinogen. Specifically, parietal cells secrete hydrochloric acid. And then these two things kind of play hand in hand to activate pepsin to begin breaking down these polypeptides. So the pepsinogen that's secreted, remember this suffix gen is indicating that it is in the inactive form. That must be activated by the presence of hydrochloric acid. So hydrochloric acid, and to a lesser extent, already existing pepsinogen. So basically, if you've got pepsinogen that's already present in the stomach, that can activate pepsinogen to pepsin, but hydrochloric acid is the main substance that converts pepsinogen to pepsin. So basically this pepsinogen becomes activated to its active form, pepsin, and then that is the enzyme that begins working on these peptides, breaking them down into smaller fragments, okay? So the chief cells are secreting the pepsinogen in its inactivated form. The parietal cells are secreting acid, and then the acid is what is activating the pepsinogen to pepsin, okay? I hope that's clear. Okay, let's talk about what happens in the small intestines. So in the small intestines, we have another group of pancreatic proteases that are being secreted from the pancreas. So remember we talked about that pancreatic duct, which opens into the duodenum here. And this is where those digestive juices that are containing these pancreatic proteases can enter the lumen of the duodenum. So the proteases are trypsin, chymotrypsin, and carboxypeptidase, and they are actually released again in their inactive form. So they're released in the form of trypsinogen, chymotrypsinogen, and procarboxypeptidase, which are not active. And trypsinogen is the main protease that is going to be activated by enterokinase. So enterokinase is an enzyme that is hanging around here in the brush border of the small intestine, and it will activate trypsinogen only to its active form, trypsin. Trypsin will consequently activate the other proteases. So trypsin, once it's activated by this enterokinase, will then go on to activate chymotrypsinogen to chymotrypsin and procarboxypeptidase to carboxypeptidase. And then all of these activated enzymes will continue the breakdown process of any protein that is coming through this part of the lumen. So essentially the stomach is kind of getting things started with pepsin, starting the breakdown of those large proteins to smaller peptides. And then here in the small intestine, these are the pancreatic proteases coming from the pancreas are kind of finishing that breakdown process. Chymotrypsinogen, which is converted to chymotrypsin, procarboxypeptidase converted to carboxypeptidase, and then the main one that's that's activating the others, which is trypsinogen, and its active form, trypsin. Okay. All right. So then our amino acids, so at this point we've got amino acids. So now we're breaking down those peptides into their smallest component, amino acids, and they can be uptaken or absorbed into the bloodstream across the epithelial cell via two mechanisms. So across the apical surface, they're going to be absorbed via active transport, secondary active transport. So think about this mechanism, although this is saying glucose, think of this as being amino acids. And so this is the mechanism by which amino acids are uptaken into the epithelial cell. And then they're going to be absorbed across the basolateral surface by facilitated diffusion. So across the basement membrane, they won't require any energy, but it will require a gradient and a transporter. Okay, so facilitated diffusion. And that is for amino acids. For peptides, the smaller peptides, dipeptides or tripeptides, they are uptaken into the cell actively, same way as the amino acids are, via this co-transport or secondary active transport. And then when they are in the cell, they are then broken down to amino acids, and then those amino acids can be absorbed across the basolateral membrane, again by facilitated diffusion. So basically, amino acids or smaller peptides can be absorbed across the apical surface via secondary active transport, but then only amino acids. So even if you absorb those smaller peptides, once they're in the cell, they must be broken down to amino acids, and then amino acids can move across the basolateral surface via facilitated diffusion, which, if we remember, facilitated diffusion doesn't require energy. It's going according to the gradient. So we've got lots of these amino acids in the cell, no amino acids in the bloodstream here. And so we've got a gradient, but we do require a protein transporter to move them according to that gradient.